A very good morning to everyone. Today I am here to present my graduate seminar entitled Evidence for the Loss of Plasminogen in Receptor KT gene in chicken. One of the most fascinating aspects of nature is the diversity of life, and evolution is the mechanism that produces this diversity and adaptation. And if you think about it, does gene loss have a role in it? During evolution, genes can be created, get mutated, duplicated, or even get lost. And losing genes are often detrimental for an organism because they are associated with malformations and diseases. However, there is a popular hypothesis in evolution called less is more hypothesis, which proposes that losing a gene can be beneficial for an organism. Despite being somewhat counterintuitive, there are examples that gene loss contribute to adaptations. For example, loss of hair genes in dolphins and whales. As we know, dolphins and whales are mammals, and one of the main characters of mammals is their body hair. However, dolphins and whales lack body hair. Their ancestors were these land dwelling mammals that had the hair to keep them warm. However, when they transitioned from land to water, hair can no longer serve as a insulator, and its presence would actually slow swimming. Hence, lack of hair become an adaptation to the aquatic life, making them more efficient swimmers. Now, how does this happen? For a gene to be truly lost in a species or lineage, the change must occur in a DNA sequence, and it can happen mainly in two ways. One is abrupt mutational events such as recombination and transposition, which removes a larger region of DNA, and second is a slow process of accumulating mutations. These are small changes, but they occur in a very important sites. For example, you can see a gene here as a reference. Here you can see this gene as a reference, and there is a substitution that made a premature stop codon, and insertion or deletion which shift the frame of this gene, and there are mutations in the splice sites which affect the splicing events, and there are mutations in the start codon. Now the question comes that why to study gene loss? If you look at the literature, there are great attention has given to the origin of new genes and gain of functions, but gene loss has been entirely ignored for a long period of time. This is because natural selection is typically thought to mostly favor gain of function mutations, which increase an organism's fitness, while removing removing loss of function mutations which are considered deleterious. However, as we have seen in the example of dolphins, there are several other cases in which uh, loss of function mutation can proven as a strategy for an adaptation. Hence, to better understand the evolution, it is critical to look at gene loss, and confirming gene loss events of functionally important genes can pave the pathway for functional studies, and it also have biomedical implications if these genes are associated with immunity and diseases. Now, to identify genes, there are several challenges. The identification of gene loss is done mainly using bioinformatics based tools. For example, in birds, there are several genes reported to be lost, especially in chicken. Example, adipone carrying genes. Lack of those sequences where because those gene sequences were missing in their genome, it was initially served as an evidence for the loss. However, later it was found that many of these genes are not truly lost. Rather, it was because the genome of the birds are in general more compact, and the inability to find those sequences due to the limitations in properly sequencing their genome, uh, and there are still several genes that are missing and yet to be found, attributing to challenges to finding gene losses. There are mainly they are mainly the limitations of sequencing. The challenges of gene losses are mainly the limitations of sequencing, which are gaps in the genome, which is the absence of sequencing reads at a particular location. So you can see here, long read sequencing can, to an extent, can tackle this problem. However, several species, most of the species don't have this long read sequencing data, and it remains as a challenge to find gene losses with gaps in the genome. And high GC content also makes it very difficult to sequence some part of genome, such as repeat regions, and lack of conserved gene synteny and RNA expression data makes it very difficult to establish that a gene is truly lost or not. Now, about these adipokine genes that were missing, there were 5 functionally related genes reported to be missing in chicken, and these adipokines, they are TNF alpha, leptin, PAI1, omentin, and resistin. These are cytokines secreted by adipose tissue and play a major role in inflammation and insulin resistance. Hence, they are not likely to be truly lost, and subsequent studies showed evidence that they're actually present in birds. So, TNF alpha and leptin were found in chicken, and PAI1 and omentin was found in other birds. Now, one of these genes called PAI1, plasminogen activator inhibitor, is a plasminogen receptor, which is an important component of a blood coagulation system, and it is still not found in chicken. Now, if we look at the blood coagulation system, at its core, it's eventually the interaction. It's just an interaction between three molecules: fibrinogen, thrombin, and plasmin. During clotting, a fibrous soluble protein called fibrinogen becomes active fibrin, which is a clot maker, and the process is called coagulation. And fibrin is and this fibrin is activated by a protease called thrombin. So the balance between both coagulation, forming clots, and fibrinolysis is required for
Yeah, now
proper
it's fine.
homeostasis. Okay. So in case of birds, they form clots very slowly compared to mammals. So this prolonged clotting time is because they don't have platelets. Now, if we look at the evolution of blood clotting in vertebrates, a proper coagulation system first appeared in early vertebrate, and in the early vertebrates like fish, amphibians, reptiles, birds, they had thrombocytes that are spindle shaped, and they were nucleated. These are the first cells to evolve to specialize in homeostasis, in hemostasis. Sorry. Then later, uh, megakaryocytes have emerged, which are giant precursors which fragments to form non-nucleated platelets. Mammals have evolved platelets, and these platelets are thrombocytes that are equivalent to other non-mammalian vertebrates. Although the evolutionary path that led to this origin of platelets in mammals is still a mystery, it has proposed that platelets have several advantages over other thrombocytes, such as faster aggregation, and they have several other roles uh, which might explain why they have evolved. And there are various proteins such as plasminogen receptors known to play important role in hemostasis are present in this platelets. We have seen the role of pre-AI1. Now, if you look at the PLGRKT, which is another plasminogen receptor on platelet, now this this PLGRKT is a recently discovered receptor called plasminogen receptor with a C-terminal lysine, and it activates plasminogen activators such as TPA and UPA via C-terminal lysine to promote fibrinolysis. And as you can see, in its predicted structure, it is an integral membrane protein with a transmembrane domain, and a main characteristic is its exposed C-terminal lysine, which interacts with the plasminogen activators. It has contrasting functions with PAI1 receptor. PAI1 promote clot stabilization, as you can see here, uh, by by blocking the fibrinolysis, and PLGRKT promote fibrinolysis by activating the plasminogen activators. And both receptors are required to maintain hemostasis. And additionally, PLGRKT has other established roles in mammals, mainly in immunity, such as regulating macrophages, which is also involved in efferocytes and efferocytosis, and cell invasion and migration require extracellular matrix degradation, which are also done by this proteases plasmin, and they're also involved in lactation. In the PLGRKT regulates the composition of extracellular matrix in lactation and forming the ducts in the glands. Now, the PAI1 gene, which is one component in this regulation, has not been found in chicken, while the other component, PLGRKT, its function has never been studied in birds. And this established role of the PLGRKT uh, provides an evolutionary context in which we can look for gene loss in other birds, especially birds. Since birds lack platelets, it would be uh, an interesting scenario to look for their presence in birds. Now, to identify the loss of a gene, there are several steps which form a workflow of checkpoints. First, we can search that gene in its genome, take the gene we want to check from a closely related species, and then search it uh, in the target genome using BLAST. BLAST is an alignment tool which is this is the most commonly used search tool, and it's proven to be very uh, fast and accurate. And if the gene is not present, if the gene is present, it will give a match, and if it is missing, that doesn't mean the gene is lost. So in this figure, we can see the middle yellow colored boxes are the main steps, and the left side are the inputs, and the right side we have the outputs, and the tools used are marked in red. So if it is missing, that doesn't mean the gene is lost. Since the genome can have assembly errors, we can look at the unassembled raw sequencing rates from several sequencing experiments of the same species. So in that way we can tackle the errors in the assembly. And next, it's not just the presence of the gene, its location is also a true indicator of its identity, which means that the order in which a set of genes occur in a genome would be same between closely related species. So this conserved block of genes is called Synteny. And gene Synteny based identification of gene remnants is generally considered as strong evidence to establish gene loss. Now, to ensure the gene is lost, next we check whether it is expressed in any tissues, and finally we look at the phylogeny, whether this detected gene loss events are shared between closely related species, which would further provides the confidence of these loss events. So, so we looked at chicken. Since loss of gene with conserved synteny is considered as the strong evidence, first synteny was checked. To check synteny, the genomic regions flanking the PLGRKT gene was extracted. Several genes upstream and downstream was extracted from several vertebrates, and it was found, as we can see, there are several mammals which are colored M, and reptiles and amphibians and even birds. Synteny regions were extracted and aligned, and it was found that the synteny is conserved. So the synteny of PLGRKT gene is conserved across mammals, reptiles like gecko, alligator, and rodents, and birds like duck. However, uh, in chicken, all the genes were present except for PLGRKT, which suggests that suggests a gene loss event in chicken. Now, to check exactly what happened in PLGRKT, uh, checked the genome of chicken. The chicken genome was PLGRKT have 4 exons, and it's found to have first and second exon is completely lost, and third exon was partially lost, and fourth exon was completely intact. And in addition to chicken, uh, checked other families of galliforms. Galliforms is the order in which chicken comes under in the phylogeny. All the 5 families of this galliforms taken a representative species and observed that this exon 2 loss is shared between all these families. And exon 1 and 3, partial loss of 3 are observed in Phasianidae and Nimididae. In this expanded view, we can see the exon 3 and exon 4, and here you can see the partial loss of exon 3 in the 6 species of galliforms at the bottom. Now, to ensure this loss and these changes, unassembled raw read sequence data were also checked for these galliforms, and in the raw reads also we observed the same changes that we observed in the genome. To further validate, we check long sequencing reads. So the identification of these changes from the short, even from the raw read, has some limitations. such as lack of long-range accuracy of the genome. So, to avoid such biases, we validated the correctness of the genome using long sequencing read data made from PacBio, and when when reads longer than 10 kb were mapped to the PLGRKT gene of chicken, we could observe that the entire PLGRKT region, all the way up to the flanking genes on both sides, were spanned by these long reads without any gaps, which supports that the long range correctness of the chicken genome and the changes and gene loss events that we identified in the sequence are reliable. Now, as we know that there are such large disruptions occurred in chicken genome, chicken PLGRKT gene, to confirm that it is actually gene loss, next step is to check whether the gene is expressed or not. Spleen and gonads are the two tissues which showed high expression of PLGRKT in duck. Duck is an anseriform, which is a closest order to the galliforms, which contain chicken. Now, if you look at this box, the columns are exons, represents exons, and the rows represents the tissues. And these gray lines represent an expressed RNA molecule. If we look at duck and the anseriforms, can see the PLGRKT, all 4 exons are expressed, and if we look at galliforms, none of them are expressing, none of the exons are expressing, showing any reads of RNA in these 2 tissues. And additionally, checked 20 different tissues from chicken, and which are mentioned here, and none of them are shown any expression of PLGRKT in those tissues. This confirms that the gene PLGRKT is lost in chicken. Now, reconstructing this evolutionary timeline of galliform-specific gene loss events, it provides a framework when and how these events happen, and approximating this time is based on species splitting time and gene presence or absence. It basically requires a high quality genome and a time calibrated phylogenetic tree containing a species with gene loss and an intact gene. So here there is a galliform which has lost the gene, and this first 12 species are galliforms and last 3 are anseriforms. And here their exon status are also shown. So yellow means partial loss, green means the exon is intact, the red means it's missing. So based on the prevalence of shared losses, we can see that exon 2 is lost around 68 to 80 million years ago, because the exon 2 is missing in all the galliforms and it is present in anseriforms, which gives which puts it in an upper bound of 80 million years ago, and it occurred before the branching of the first species, which suggests that this event must have happened sometime in between 80 and 68 million years ago. And similarly, timeline is further resolved of exon 1 event and partial loss of exon 3. In addition to this dating, we could also see that this gene loss event happened through a sequence of events starting from exon 2, which is shared between all the galliforms, then exon 1, and finally the partial loss of exon 3. Now, for a functionally important gene to get lost, it should be dispensable, which means there is an alternate pathway or a redundant gene which can compare, which can compensate for its loss. So when a gene is dispensable, it can afford to accumulate more changes in this sequence. So uh, it's not just in chicken. We looked for such kind of signatures in all the birds which have high quality genome, raw reads, and RNA sequence data available with a conserved Synteny. And screening all those genomes, we found 9 independent coding frame disruptions in different lineages. Here you can see they are marked in red. For example, event 1 is occurred in crows, which is this colors represents the exons, and the shape of represents the kind of change, whether it's an insertion, deletion, or a complete missing. So, for example, in event 1, there are 2 deletions occurred, which led to 6 stop codons in the gene. And since it occurred early in the phylogeny, it is present in all of this 10 crow species. And similarly, there is event 2 and 3 occurred in finches. Event 4 occurred in 10 passeriform bird species. And 5, 6, 7, 8 occurred in different species such as woodrigger, flycatcher. And the event 9 occurred in falcons. So these changes are not just mutation; they all are disrupting the coding frames. Hence, they are gene loss events. We have checked its RNA expression, its Synteny. All of them were suggesting that this gene is not expressed in all these species which had this coding frame disrupting mutations. And this several of them occurring independently in different lineages suggest that this gene is in general dispensable, and uh, we can say that this gene is must have undergone several mutations to and eventually resulted in complete loss. And to conclude, gene loss can act as a major engine of evolution uh, by being an adaptive by being an adaptation for the organism. And in vertebrate blood clotting, uh, there were several modifications occurred during the evolution, and one of the major gene involved in the maintaining homeostasis is PLGRKT gene. It is we found the evidence that it is lost in chicken. By this following evidences, the complete loss of exons 1, 2, and partial loss of exon 3, and a lack of RNA expression in 22 different tissues, and a conserved Synteny with just the missing of PLGRKT gene. and the shared loss events across all the families in the galliform order. And this loss of functionally important genes suggests that the gene is dispensable, and it is supported by the gene loss events in uh, other birds, which non-galliform birds, and we have observed 9 different independent events in different lineages, which uh, led to the loss of PLGRKT gene. And this gene loss in galliforms uh, and the strong support for this independent losses provides a natural knockout that will result in better understanding what is its functional roles. And, finally, I would like to acknowledge Dr. Nagarjun Vijay for his constant guidance, IISER Bhopal for facilities, and all the CEG lab members for their healthy discussions. Thank you.